So we'll have the very first presentation by Sharon Na Ayele Aite. Now, Sharon is a second-year PhD student of the Department of Clinical Microbiology, and her project has been sponsored by GWOC. Sharon.
Good afternoon, faculty members. Good afternoon, colleagues. As rightfully mentioned, I'm Sharian Aite, and my presentation is titled Zoonotic and Potentially Zoonotic Viruses in Game Animals and Livestock in Ghana. And I would like to put across that this work is still ongoing. Okay, this will be my outline for the presentation. So to begin with, about 60% of emerging and re-emerging infectious diseases affecting humans are said to have zoonotic origin. And for the past decades, the high-impact pandemics and outbreaks which has affected humans have been as a result of introduction from livestock and wild animals as reservoirs. Also, livestock that live in close proximity to humans can facilitate the transmission of zoonotic viruses through the wildlife-livestock-human interface. And there are various techniques available for the detection of these viruses, some of which are the classical cell culture, PCR, serological assays, and next-generation sequencing. With the exception of next-generation sequencing, the other methods require an individual having a prior knowledge of similar viruses. So figure 1 depicts the various transmission routes of zoonotic viruses, with the common route being the middle route, where other game animals such as migratory birds, bats transmit the viruses they harbor to other game animals. And when humans and domesticated animals have an interaction with these animals, they stand a chance of having been infected with the viruses these animals harbor, leading to a potential human-to-human transmission. So due to the high demand for animal protein, there has been a major expansion in the commercial animal farming, which has led to major disease outbreaks and pandemics, such as the recent SARS-CoV-2, Ebola, and Marburg. Given this, it is therefore important to identify known and unknown viral pathogens harbored in this animal, which can lead or aid experts in tracing the origins of specific epidemics, as well as predict the potential sources of future outbreaks, which can help implement policies to manage or prevent these pandemics from happening. So my main objective is to perform a virus metagenomic investigation to identify known and unknown viruses harbored in these wildlife and livestock. To achieve my main aim, I would first identify the zoonotic and potentially zoonotic viruses harbored in these tissues, after which I would investigate their genetic diversity and evolutionary relationship with reference strains. Further, I would determine the risks wildlife pose and their potential contribution to spread of zoonotic viruses along the bushmeat trade. Further, I would also investigate the prevalence and genetic diversity of astroviruses in archived fecal samples from Eidolon helvum in Ghana, i.e. fruit-eating bats. And finally, I would investigate the prevalence of paramyxoviruses in archived tissues from livestock in Ghana. So for my objective 1 and 2, they are hand in hand. So I would use an exploratory cross-sectional study for which approval has been sought from the Animal Research Ethics of KNUST and the Wildlife Division of Ghana Forestry Commission. I would be taking tissue and gastrointestinal samples from these bushmeat that are preferred for consumption in Ghana. And my selected study sites are the less explored bushmeat market in Bunu East, Volta, Oti, and Greater Accra regions. So figure 2 depicts the community entry into Techiman and Amejope in the Volta region. So this is my study procedure for objective 1 and 2. After community sensitization, a short structured questionnaire would be administered to the handlers of these bushmeat, after which, with the help of a veterinarian on site, we take portions of organs such as the heart, liver, spleen, and even fecal matter. And in the lab, we would extract viral nucleic acid from these samples and then screen them for major zoonotic viral families, after which the positive samples will be sequenced using the mini-seq platform, and all negatives will be pooled and sequenced using a high-throughput sequencing platform. Figure 4 depicts the overview of samples I have taken so far and from the regions I have sampled from. So far I've sampled from Oti, Bono East, and Volta region, with Bono East having the highest number of bushmeat sampled, followed by Oti and Volta. But Oti has the highest diversity compared to the other regions. And the highest number of species sampled is the grass cutter, followed by the ground squirrel, and pangolin being the least. This gives an overview of the samples I have taken so far. Also, Bono leads this chart, followed by Oti and Volta. And the highest number of tissues or samples I have taken is the lungs, and the least being the lymph nodes. So for my objective 3, I would use a cross-sectional study approach. And ethical approval has been sought from the Human Ethics Committee of KNUST. I'll be taking blood samples from these handlers of bushmeat. And the handlers of bushmeat are the vendors who sell the fresh meat, those who use this fresh meat to cook, that is chop bars and eateries, and also hunters. And my study sites are same for objective 1 and 2, which are the Bono East, Greater Accra, Oti, and Volta region. And the study procedure is similar to objective 1 and 2, just that there's an introduction of blood samples being taken from the handlers of bushmeat. And then further serological analysis will be conducted on these blood samples. And this gives the overview of blood samples I have taken so far across the 3 regions. And as expected, Bono East had the highest number, followed by Volta and Oti. And we were able to sample just 11 hunters and about 97 vendors. For my objective 4 and 5, they are a retrospective study. And permission to sample from these animals were sought from the Wildlife Division of Ghana Forestry Commission. For objective 4, I'll be using the Eidolon Helvum fecal samples, and 5 will be liver and lung samples from domesticated rabbits and grass cutters. So about half of the laboratory analysis for objective 4 and 5 has been conducted, which is extraction and screening for major viral families, zoonotic viral families. We are yet to sequence the positives and the negatives as well, then analyzed using bioinformatics. So this is the overview of samples, the results from gel electrophoresis. I screened about 158 pools of the Eidolon bat fecal matter. And out of these, 3 tested positive, representing 0.02. And for the liver and lung samples from the domesticated rabbit and grass cutter, none of them tested positive for the paramyxoviruses. This is also a representation of the gel electrophoresis results. So the required band size of the astrovirus is around 438 base pairs. And you could see the bands in the red box. They are around 438 base pair. If you map it to the positive control, you could see these 2 are positive. And they were sent for Sanger sequencing, just to confirm if it's actually astrovirus, and they are. So I'm yet to resolve the pools, then sequence them to know how related or distant they are from the reference strains. So my next step is to sample from my last study site complete my laboratory analysis and data analysis. So at the end of the study, data from this study will provide public health experts with the necessary information to prepare or prevent for the next pandemic or outbreak. Also, data would help policymakers and stakeholders implement policies related to animal trade, vaccination, and infection control. These are my references. I would like to acknowledge the following institutions. Thank you.
Thank you very much, Shahian, for this. What we'll be doing is that we'll look at the first three presentations, and then we'll take questions and comments, please. So we'll listen to Georgia J. now on the molecular epidemiology immunological responses to SARS-CoV-2 and other respiratory viruses in selected urban and rural areas of Ghana. Now, Georgia J is a third-year clinical microbiology PhD student. He's also being funded by GWOC. George.
Good afternoon, faculty and fellow students. My name is George Ajay, a PhD third-year student at the Department of Clinical Microbiology. I'm here to share with you my work entitled Molecular Epidemiology and Immunological Responses to SARS-CoV-2 and other respiratory viruses in selected urban and rural areas of Ghana. Below are my supervisors, Drs. Michael Owusu and Philip Eldia, and Augustina Silverkin, professors Yao Adusa Akodie, Richard Odame Phillips, and Christian Drostin. These will be my presentation outline. You and I are aware of the current state of COVID to the barest minimum. And the graph you see depicts what is happening now as of 2023. And WHO this year, 2024, released this for us to know the state of the cases and the death rates, which are very minimal. Also, COVID-19 vaccine administered as of last year, 31st December, also shows in the first map, showing the regions with the color intensity from light green to deeper green and then very faint. The faint one shows that the number there being vaccinated is very low, followed by the others and then the deeper ones. The other map also shows the various district distribution and the vaccinations given. During the pandemic, measures employed in Ghana to avert the effect of COVID-19 included non-pharmaceutical interventions and vaccination campaigns. These measures were done to prevent the spread of the virus from persons to persons so that we can contain it, since it was killing people and knowledge about it was very low at that time. The uses of these interventions have not been fully evaluated in the population. The measures we employed, we want to find out how useful it was to help curb the disease in our setting now. Therefore, they need to establish the disease exposure and immunity acquired in different socio-demographic contexts in Ghana, meaning that we want to find out within different places in Ghana, the urban and the rural areas, how it was felt during the pandemic. We want to find out how the disease, people contracted it, how immunity was developed, and how the immunity will wane, or the longevity of this immunity. And also find the difference in seroprevalence between the urban and the rural. Owusu et al. 2014 also let us understand that acute respiratory tract infections represent an important public health concern worldwide, and as observed in the recent pandemic involving SARS-CoV-2. Also, Lewis H. et al. Also 2022 also inform us that there is a limited data on the genomic epidemiology of SARS-CoV-2 and other respiratory viruses in many parts of Africa, which is true. Ghana experienced the same thing during that time. And as the figures were changing, our laboratory was not even up to beat to test for PCR and the rest. Beginning, I think, KCCR and then Noguchi was helping us with the testing, and other labs were brought up. This shows that there's a lot of work to be done to face future pandemics, the need for the study, and all of us should be aware of what is happening. The specific objective of my study, one, is to determine the prevalence of SARS-CoV-2 and other respiratory viruses in selected areas in Ghana. Two, to determine the disease exposure and COVID-19 vaccination rate between the urban and the rural areas. Then to determine the socio-demographic characteristics, risk factors of SARS-CoV-2 and other respiratory viral infections at selected study sites. Then lastly, to determine the molecular epidemiology and geospatial distribution of SARS-CoV-2 and other RVs at selected study sites. Of all these 4 sites, I have data to support all, but I would like to share with you objective number 2 results. And the first, third, and fourth are still in process because it is linked with the viruses that we are testing. All those samples have been extracted and we are at the PCR level. So I'd like to share with you the ELISA results for the disease exposure and the vaccination rate within the country. For the materials and methods, these are the areas so far I've visited for sampling. Tamale and Kumasi being the urban areas, Obuasi, Forikumu and Boyim forming the rural areas. And then this, the meeting with community leaders at Tamale. Then for the ethics for the study has been sought by Committee for Human Research and Population Ethics at KNUST and Ghana Health Service. And then the study design is cross-sectional and longitudinal. Study sites I've mentioned already, rural and urban areas with the towns mentioned. The study population will be whoever present with or without symptoms of COVID-19. And the inclusion and exclusion criteria will be, the inclusion will be ability to give consent and if you are more than 10 years or 10 years. And the exclusion will be history or peace status. The procedure for the two testing or the various methods will be recruitment of participants and then with questionnaire administration, using red cap to pick the sample. Then the sample collection will be blood sampling and nasopharyngeal sampling. Then this will be done with RNA extraction PCR for the swabs, and then the blood will be done with ELISAs. And the ELISA are 3 full steps that I will explain further. Then the data will be entered and the analysis and bioinformatics will be done. Here's a picture showing the ELISA and then RNA extraction in the lab here at KCCR. The results showing up that the number of participants so far from Kumasi who have enrolled are 479, Obuasi 209, Forikumu 207, Boyen 180, and Tamale 481, meaning Tamale has more participants. And the age distribution of participants are below, ranging from 10 to 80 years, with 10 to 20 having the highest figure. With 10 to 12 having the highest figure. And then the gender distribution of participants, having more females being enrolled than males. So it's house-to-house based study, and when any house you enter, you talk to them, you explain the study to them. Some will consent, some will not consent. At the end of the day, we had more females being enrolled in the study. And that is the results of the sample sites that we have, and the urban have similar with the Kumasi and Tamale, the swabs and the serum and the rural areas following. For objective number 2 results, so far vaccination status that we found 54% responding yes to vaccination, 54.7 responding yes to vaccination, and 45.3 responding no to the vaccination. And this is the distribution of various vaccine types that we collected, or we use the vaccine card to collect that info for AstraZeneca, Johnson & Johnson, Pfizer, and Moderna. Of all these, AstraZeneca is leading 48.2% of the type of vaccine that we receive in Ghana. You and I are aware that the beginning of the pandemic, we all received AstraZeneca. And the results I'm going to also talk about will let you understand that most of us are either were infected or most of us are vaccinated with the vaccine. For the serum samples that were tested, Kumasi, we had 383, Obuasi 84, Boyim 131, Forikumu 131, Boyim 137, Tamale 360s. The reason for this is that most participants will give you the swab all right, but they will object to the blood giving out. You need to talk to them, explain more. However, we managed to get 1,001 participants who willingly consent to be part of the study. And we have done anti-S ELISA, that is the spike protein of the virus. We've done ELISA on that, and the results are seen on the table, with 98.2% of the total 1,001 being positive, showing that all these participants were exposed to the virus or have been given vaccination. And with the distribution, as you see at the end, Kumasi, Tamale, Furukum, Boyem, and the rest with the percentages up, as you can see, with Tamale leading with 36.5%. Also, we continue with objective 2 results showing the anti-NCP ELISA result, the nucleocapsid of the virion or the SARS-CoV-2 virus. And now you only elicit response to this when you have natural response. The spike is where you have both natural response and then maybe exposed to a vaccine. And with this ELISA testing, we also came out with this result showing a positive rate of 47.8 for 477 participants and a negative of 52.2 for 521. Of all these, you also match the results to see X only, those who have an S only 98.2, and those having both the NCP and S as 47.8. And we'll follow up on all these participants to know how long their immunity lasts or the antibody titer will last, and also we will check and see if there's any seroprevalence between the urban and the rural. The next step for the study will be to we observe a high seroconversion, that is the IgG, in all the study sites, though yet to be confirmed with neutralization assay respectively. This means that the positive we are seeing, we have to go further, do ELISA testing, which is called neutralizer, for specifically SARS-CoV-2 in question, so that we'll know that the positive for the IgG and the NCP are true positives. So when you confirm that those positive will be used for the study. Neutralization, I say, simply means having the neutralizing antibody that will inhibit further infection from the same virus that you were infected, or the same strain that you were infected. All ELISA confirmed positive persons will be followed up after 1 year to know the difference in seroprevalence between the urban and the rural. Further testing will be done on the remaining samples to estimate the prevalence of SARS-CoV-2 and other respiratory virus circulating in Ghana by multiplex real-time PCR, where we will be testing for respiratory viruses by REST panel. Expected outcome of this study will be information gathered will be essential for policymakers to prioritize health strategies and assess the effectiveness of public health measures implemented during the pandemic. And from this study, we can get informed knowledge and data to be implemented so that it can guide us into the future for future pandemics or future diseases that will crop up. Findings will provide useful baseline data that will inform further molecular epidemiological studies on SARS-CoV-2 and other respiratory viruses in Ghana. So grateful to these institutions for funding exploratory work. Thank you.
Thank you very much, George. Thank you, George. Like Prof said, we would take a third presentation and then we would have questions on these emerging infectious disease pathogens. Our next presentation will be by Theophilus Odom. So Theophilus is a veterinarian and his PhD is in One Health with the School of Veterinary Medicine at the KNUST here. He's in his second year. Theophilus is presenting on, let me just check again. I hope there are no changes in your topic. But as stated in the book, Theophilus is speaking to us on the status of Marburg virus in bats, domestic animals, and dogs in Ghana. Theophilus, the floor is yours.
Good afternoon to you all. My name is Theophilus. I'm a veterinarian at the School of Veterinary Medicine. I'm a second year PhD student, and I'll be talking to you about the status of filoviruses, specifically Marburg virus, in bat and domestic animals in Ghana. This is my outline of my presentation. We all know that in 2022, when Ghana was coming out of the wounds of COVID-19, we heard about Marburg outbreak in Ashanti and the Western region, precisely Formina and Bogoso in the Western region. Then the whole country was thrown into a state of shock. We decided to investigate about the Marburg, where it's coming from, what is the status. We realized that Marburg and Ebola virus are sister virus belonging to the family Filoviridae. And we know also that the Marburg is or can be found naturally in Egyptian rosette bat, which is also a common fruit bat in some part of Ghana. Marburg is restricted or has been restricted to East Africa, precisely Democratic Republic of Congo, Kenya, Angola, and some parts of Uganda. But it is not known in West Africa. The first case in West Africa happened in 2021 in Guinea, Equatorial Guinea. Then in 2022 we have Ghana. 2023 we have Equatorial Guinea. So it is spreading in West Africa. But what is the problem now? The natural reservoir has not been fully studied in Ghana because we don't know where the first case of Marburg occurred from. The role of domestic and livestock animals and pet dogs are not also known or not documented. What are also some of the socio-ecological conditions and risk factors that expose human to the Marburg virus? We don't also know. And there's also a limited number of serological assays, which means that the assay to diagnose Marburg serologically is not available commercially. So we have to investigate the first outbreak of Ghana, which appeared that it is not known in Ghana. We don't know where it came from, whether it's coming from the bat directly to human or to domestic animals activities. And also studying this virus in the natural reservoir will also give us a broad spectrum of information so that we can plan as a country knowing the transmission cycle from animals to human. So my main objective for this study is to determine the presence of Marburg virus in bats and also the antibodies in livestock and dogs in some selected areas in Ghana. Then specifically, we will determine the presence of the RNA in tissues in bats. We also develop and establish a serological assay, which is the immunofluorescent test assay for bats, livestock, and dog testing. Then we also investigate the possible exposure or the spillover of the virus to livestock and dogs, and also we also assess some of the socio-ecological conditions and risk factors associated with the Marburg outbreak in selected areas in Ghana. So with this, we selected 5 areas based on existing data. The first outbreak appeared to have come from Ashanti region and Western region. So we have Fomina and Kasi Kwamine area. Then the Western region is Pristia-Huni Valley. Then Volta region, Vegolokwati. Vegolokwati is area where previous studies have shown that the Egyptian rosette bat is found in some caves in these areas. Bono East, Mueyem Enclave, and Tanoboase is also an area where we have caves where these bats are encountered or can be found, or they roast. Then Savannah is due to the human and animal-wildlife interaction. There are also some caves around that it is believed that these bats are hiding in these caves. So work done so far, we've done some field work in Ashanti and Western region. Unfortunately, in Ashanti and Western region, we did not find caves there, but we were able to trap bats in the forest where galamsey mining is occurring. We found a lot of Megachiroptera bats and Microchiroptera bats. Unfortunately, we didn't get Egyptian rosette bat in Ashanti and Western region. But in Boyem, in the Bono East region, in spending 2 weeks there, we were able to capture 43 Egyptian rosette bats from caves around the Boyem enclave. So this picture depicts some of our work in the field, how we capture the bats, how we were able to select the tissues, organs from them. Then the livestock and dog field samples, we were able to cover our study site. But we were able to do it in dry season and wet season. So in the Ashanti, Western, Bono, Volta, and Savannah, we were able to sample all these animals from these regions. In the wet season, we did April to June, and the dry season, we did August to October. Then we established our PCR assay. Our PCR assay is a one-step RT-PCR assay which has been established, validated, and optimized for Marburg work in Charité Lab in Germany. Then we tested the first samples from Ashanti and Western region. Unfortunately, we did not get any positive from the samples that we tested. It is true also because we didn't get Egyptian rosetus bat from these areas. We were able to establish as an objective a serological assay, which is the immunofluorescent test assay, IFT. This consists of designing primers, doing enzyme digestion using the PCAS vector and expressing it in Vero B4 cells. So it has been established, validated, and optimized in Ghana. I did the test in Germany, but we have established it in our laboratory, Accra Veterinary Laboratory in Accra. And the result is impressive. We've been able to use to test some of the samples from 43 bats that we harvested from Buayem in the town of in Buayem in the Bono East region. And we found some positive antibodies reacting to the assay that we have. And you can see from here, you can see some of the green, brilliant cells over there, showing positive cells in fusion with the sera from the bats. So our results shows that the immunofluorescent assay for the detection of the Marburg virus antibodies for bats, livestock, and dogs has been developed and it has been established, it has been validated, and it has been optimized to be used in Ghana. And 12 out of the 43 Egyptian rosetus bats that we tested shown positive with our assay, and all the 239 bat sample, non-Egyptian rosetus bats, tested negative with our PCR assay. For our research work has a policy implication because the information from the design of the Marburg virus will help our surveillance system in Ghana so that we can incorporate in our surveillance for the animal sector, and also the human sector can also use the assay. And also we implement a strategy on how and when we should also target our surveillance activity. And also establishing serological diagnostic assay for detection of Marburg antibody in livestock in dogs is very essential because it will also reduce the cost and also searching for commercial assay which is not existing in the world. Our next step is to continue field bat sampling in the Savannah and Volta region, even though it's very expensive, but we have to complete it. And also complete the IFT testing for the remaining samples. It's very tedious because you have to prepare the slides and spend time observing the slides. It's tedious, but it's much easier than to rely on commercial assay, which is not in existence. And also to be able to submit our first manuscript by August 2024. Thank JWAC, DAD, VSD, WHO, who sponsored part of the work, and Charité and KNUST for this work. Thank you very much.
Thank you very much, Odun, for this. I guess the first three speakers have all been on time. We've not had the opportunity to stop anybody, because they've been out of time. So congratulations. We will take any comments and questions. So please, for the first three presenters. So if you have any comments about their presentation.
Any questions for the presenters? Right. We'll take in batches of 3.
Thank you so much. So I'm called Joseph Atawana. I have from the School of Veterinary Medicine. I have 2 questions. The first question goes to the
first, let us know who the question is directed.
So the first question goes to the first speaker. I want to find out whether the time of collection of your samples from the slaughtered animals will affect the ability to detect those viruses. I don't know whether I should come again or you're okay with that. Yeah, so all I'm saying is you took your samples from slaughtered animals. How long ago were those animals slaughtered? Because if it was fresh, or if it delayed, may have effects. I don't know. I'm not a virologist. Good. And secondly, to Odom, the third speaker, why have you left out cats? I saw dogs. I didn't see cats. I don't know the reason.
Thank you very much. Are you a lover of Joseph meat? All right. Second question.
Thank you very much. I think great presentation from all the presenters. My first question is for George. Your criteria for blood sampling, you indicated that you're taking from individuals who are more than 10 years old. Yes, I just wanted to know why 10 years? Why the specific 10 years? And then my next question is for Sherry-Ann. On your specific objectives, you were more specific on investigating astroviruses and then paramyxoviruses. Also, why those specific family of viruses? Thank you.
We'll take the third person.
All right, thank you. My first question goes to George. First of all, my question will be the method of selection, because you talked about Tema, Kumasi, which is a very large geographic area. So what method of selection did you use so that your data can be representative of the cities that you are talking about? The second question also is that you showed us the data on vaccination. Were these data based on first shots of the vaccine, second shot, or boosters? Did you consider all these three for the data that you took? And the last thing is that how do you justify the selection of the participants and the coverage? I think that's what I mentioned. So Theophilus, my question will be just one.
Let's take your next question in the next round, because it seems to me we have 8 questions now in interest of time. So if there's a next round, we'd like to ask those. Thank you. So I hope the presenters took note of the questions. George, is there a difference between, what is it, Forichrome or Ophorichrome? Where is that?
The Bonny East.
It's a Bonny East.
Yes.
Oh, okay. I thought Ophorichrome. All right.
Okay, so to Mr. Joseph, yes, the time of sampling can affect the viral detection. Okay. Sorry, Dr. Adao. I'm sorry for that. So yes, the time of sampling can affect the viral detection. So what we did was, before going to the field, we had contacts with veterinarians in the various regions. They went, spoke to them, sensitized them. So when we got there, all we need to do is wait for their call. They have samples. We go there, they send, open it up, and then we take our tissues. And to Michael, I focus on astroviruses and paramyxoviruses because they are the major zoonotic viral families. And as I said, we are screening them and sequencing the positive first because sequencing is quite expensive. We want to cut down costs. After that, all the negatives will be pulled and sequenced, saving some money. Thank you.
All right. To my friend who asks, why did we choose the blood selection? To our friend who asked, why did we, for the case who are above 10 years, that we selected for blood sampling. Studies have proven that kids' immunity is not well developed below 8 years, and in our study design, we decided to start from 10 years and above. So whoever consent from 10 years and above will be included, even with consent of their parents, since the laws of the country does not permit you to enroll a child which is below 18 years unless the consent of the parents. So those documents are there to confirm that. Then to my friend over there who also asked the method of selection for the towns. Actually, the study is to give an impression of what happened during the pandemic for the whole country. So we selected 2 urban areas, very populated, Kumasi and Tamale. The statistics given there are figures, we use the National Population Census data 2021, which shows the figures are high over there. I can't off mind give you the exact populations there, but you can best attest that Kumasi and Tamale are well represented for the urban maybe capitals. Then for the rural, we chose Obuasi, not the Obuasi city, but rural Obuasi within Obuasi town. Then Foriko and Boyim representing the northern sector, and Obuasi representing the south, and Kumasi representing the south, and Tamale representing the north. So the data we'll get at the end of the day will be accumulated and projected and make inference from that to predict or tell what really happened during the pandemic. And you also asked, did we consider all the short information? Yes. This study, we moved from house to house. But to start, and it's very sad for me to say that there are some households we ask them, were you vaccinated against COVID? Yes. Go and bring us your vaccination card. I can't find it. Even here in Kumasi, a lot of places, all the places we went. And you can see that only 54.7% were giving us yes. Most of them, even the 54.7% who said yes, if you check the data for the vaccine type, you realize that the data from the 3 shots that we had there are now up to the 54.7%. This shows that when a national campaign of such nature happens and you are giving any card, let's keep it for information delivery so that the country can move on, so that in future when there's anything like that, and we are coming to you for data, we can help get. So we pick all the 3. Thank you.
Dr. Joseph Atawana, yes, you asked why dog was included, not Joseph, not cat. Anyway, yes, dogs are pets and they are used for hunting. And the idea is that these bats are always in the bush, fruit bats. So when you use dog for hunting, the possibility of dogs getting in contact with bats or leftovers of bat is higher as compared to cats. And cats are always in the house. So it is wise, or it's also based on data that we selected dogs and not cats. Thank you.
Additional questions. My first one will be to George. I wanted to know whether your immunologic assay, were there any cross-reactivities? Which ones did you look out for, if any? There was a hand up. Yeah.
So please, my question goes to Mr. Theophilus Odum. During your presentation, you kept mentioning that you were focusing on sites for your research where the bats can be found in caves. So I wanted to ask, that was the criterion for selecting the habitat of the fruit bats straightly caves? If not, then why was a place like the campus of the University of Energy and Natural Resources neglected, which is widely known for the presence of fruit bats, which actually lives with the students on campus?
Take note of that. Can you pass the microphone?
Please, my question goes to Sherry-Ann. You made mention that in your study you'll be doing some genome sequencing, and then you considered some viruses. I want to ask, have you been able to identify some accession numbers specifically to serve as a reference genome for all those viruses you're really looking out for? And then to Mr. Theophilus, you made mention of, you had some various species of bats. I want to ask, I saw lungs and other, tissues or something for the bats. Did you harvest, kill those bats during your sampling? And also, how did you also distinguish the various type of bats? Because I saw some subspecies of bats. So how did you, give that kind of differences in that?
To the front. All right, I'm coming to you.
Thank you, Doc. My question goes to the. Okay. My question goes to the 3 presenters. During the presentation, I've not seen any of you indicating, the fact that in future there may be disease X. Now, as veterinary practitioners, do you have strategies that will combat that should there happen to be one? Thank you.
Thank you. Technical question. Let me take the hand which was here. I'll come back to you. Go on.
Yes, thank you. Thank you for the presentation. My question goes to second presenter. You said you were going to follow up on these individuals. I know you are in year 3. And looking at the time, I was wondering if you are going to take samples from these individuals again and run same and give us results or something. Thank you.
Let's take from you.
So to Theophilus, please, my question is, with the immunofluorescent assay that you developed, in the validation, did you compare to a gold standard? And if you did, what was the performance that, in terms of sensitivity, PPV, can we be confident in the assay that when it identifies a positive or a negative case, it's so?
Exactly. And that's linked to what I asked you about the robustness of your assay. So we'll take answers to these questions, and then if there's time, Prof. permitting, we might go back for another round. Yeah.
Okay, so to James, for my bioinformatics analysis, you can't get a session number now till you get your reads, you blast against a reference genome. So for I presented results for astroviruses. So that's the only accession number I've had. And when I blasted the 3 pools and did my phylogenetic analysis, I realized they had about 90% resemblance to other reference strains from Tanzania and Kingdom of Southern Arabia. Yeah. And to the second question, to clarify, I'm not a veterinarian, so I'm not a practitioner, but my results would inform policymakers in implementing policies regarding these outbreaks, vaccination, and animal control. So for now, I don't have any measures. My results will tell which measures we can implement. Thank you.
All right. To Prof, you asked about cross-reactivity and which one did I consider in my test. I think in testing for anti-S ELISA against SARS-CoV-2 with a spike, it's likely you can get cross-reactivity from the common human coronaviruses. So what we are going to do is to do the neutralization assay to firm up with the positive that we are having, that with the neutralizer, we are talking about only SARS-CoV-2. So that's the plan we are going to do. So that the other human coronavirus, such as the 229E, NL63, HKU1, OC43, will not be cross-reacting with the results that we'll get. And also SARS-CoV-2 from literature, SARS-CoV, which happened in 2003, currently if you test people, will still be having some antibodies. So we want to be sure that we are living with SARS-CoV-2, not others. Then the person who said, I'm third year, yes, we are in third year. We were doing a follow-up on only people who tested positive to the ELISA, only the blood sample, not the swabs. And to add to that, we have to start in August to January. And with ELISA, once we go to the field and come, we can run and then know the results and move on. And by then also, we're able to confirm the longevity and the difference in seroprevalence between the urban and rural to inform policy decision. Thank you.
Yeah. Thank you very much. I will take the question as it is, not in any order. I think you asked about if I killed the bat and how did I identify different species of the bat. Yeah, this is a very good question. First, we sought a citation from Wildlife Commission, which approved our protocol. And in our protocol, we need to euthanize the bat to be able to collect the tissues. And in the team, when we are going to the field, we always have bat ecologists. I'm not a bat ecologist. The bat ecologist is someone who is specialized in bat pathologist specialist. I'm able to do the dissection and able to identify all the organs. So with that one, I'm well covered. Then you asked about the University of UNE. I talked about Egyptian rosetus bat. These bats roast in caves and they go out in search of fruit at night. So they are not the normal fruit bat that you find them hanging on trees around 37 Hospital and other areas. These bats, they are nocturnal. They are always in caves, so they come out. So we base on the bat ecologists to be able to what? Sample. We can't sample the whole Ghana. It's very, very expensive sampling. So I think Unia is in which region? Bono. Yes, we have Bono East in it. We are very close. Previously, they were one region, so I think it's covered. Yes. And do you have a strategy to combat a disease? Should there be any one of them? As veterinarians, what we do is surveillance. We do passive and active surveillance in search of disease so that it doesn't happen. That is one of the reasons why we developed this assay, so that the assay can be used for surveillance purposes. We don't have to wait for disease to happen. We always do daily, routinely, passive and active surveillance. So we wouldn't wait for disease to happen. We will contain it before it even happens. Then the last question about the assay, whether it has been compared with gold standard and it has been validated. What? Yes, the assay was compared with indirect ELISA. We also done viral neutralization test, and all has been done. I can't share all the details here in this presentation, but it has been compared with all those tests, and it is highly sensitive and specific for these species of domestic animals, because it is species specific. So we have every species is specific for the second antibodies that was used for the assay development. Thank you.
Thank you very much. A round of applause for our panelists. Thank you, George. Thank you, Theophilus. Thank you, Sherry Ann. Prof.
Thank you, Yao. I think what we'll do is that we have two more presentations. Some of the presenters did not show up. So if there are any questions for these initial three, we'll take them at that point. I have just a comment to make. Sharian, in one of the slides you showed, taking a sample from some of your game life, I didn't see you marked up. And that got me worried. I mean, you are looking for potential zoonotic agents. So that for all of us who work with such pathogens, we don't know what we are going to find. We don't know what will happen. So please, 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 adopt the usual universal precautions against infectious agents when you are working with these, please. So that was my comment then. We'll take the next presentation this time from Nimbot. I think Nimbot is a second year, a third year PhD student in the Department of Clinical Microbiology as well, also being funded by GWOC. Yeah, okay. He will talk about the helminth immune modulation and its role in asymptomatic SARS-CoV-2 infection. A retrospective cohort study in Ghana. Nemot.
Okay. Thank you very much, distinguished guests and esteemed faculty members. I'm called Nembot Fogam Bryce Amel. I'm a third-year PhD student, Department of Clinical Microbiology. And this afternoon, I'm excited to share with you the preliminary findings of my research work on helminth immune modulation and its role in asymptomatic SARS-CoV-2 infections. This is part of my PhD thesis. This shall be my presentation outline. Thank you. So I'm pretty sure each of us here in this hall had once upon a time wondered why the COVID-19 pandemic did not kill so many people in Africa the way it did in Europe, Asia, in America. We quite remember during the peak of the pandemic, over the news and from reports from the World Health Organizations, so many developed countries, there were so many cases, hospitals were saturated, and there was no way. And actually, the prediction for Africa was that when the pandemic gets to Africa, we might even get extinct from the, from the predictions made by some international organizations. However, when the pandemic came to Africa, surprisingly, though it came to add up with our neglected tropical diseases, which we've been struggling with, and yet we did even better than most of the developed countries. So Africa, despite being the second world most populous continent, accounted for less than 5% of the global SARS-CoV-2 cases and less than 3% of the fatality rate. And this was quite impressive. And since then, scientists, one of the hypotheses brought about by this disease lack of exponential development in Africa is this presence of neglected tropical disease, more specifically helminthiasis. Let's say since then, scientists have been carrying out a lot of works to find out actually what happens with mixed findings. Some scientists suggest that co-infection with helminthiasis could actually exacerbate the severities of SARS-CoV-2. Some say co-infection could instead alleviate, and some reports says actually co-infection could have no potential influence on the severity outcome to SARS-CoV-2. However, my hypothesis, my stand is that co-infection with SARS-CoV-2 could actually alleviate the severity to SARS-CoV-2. My hypothesis is based on the fact that looking at the pathogenesis of SARS-CoV-2, when a person is being infected with SARS-CoV-2, the immune response triggered against the infection is basically type 1, which is more of pro-inflammatory, leading to the cytokine storm. And it is this cytokine storm which is responsible for the disease severity. So controlling the severity of SARS-CoV-2 also comes at controlling this cytokine storm. And the immune response to helminthiasis is more of the type 2, which is anti-inflammatory. Being anti-inflammatory in nature through the action of IL-10, transforming growth factor beta, and IL-5 has been known to suppress the type 1 immune response. So it is believed that in a co-infection scenario, the type 2 immune response could actually suppress the type 1 immunity, leading to a low cytokine storm, and a low cytokine storm could eventually lead to milder to asymptomatic outcomes. So these are my study objectives. So the first objective was to determine the influence of helminths co-infection on SARS-CoV-2 clinical outcomes. Secondly, to determine the immunological basis that underpins the association between helminth co-infection and COVID-19 clinical outcomes. And lastly, to determine the immunomodulatory effect of helminth antigens on PBMCs in vitro. In this presentation, I'm going to speak to the two first objectives. The third objective is still on the way. The results are not yet ready. So ethical approval was obtained for this study. We employed a retrospective cohort approach, utilizing samples from two major regions in Ghana, the Ashanti and the Upper East region. Members who presented with or without symptoms of SARS-CoV-2 were included. However, we excluded pregnant women, individuals on immunosuppressive drugs, or individuals who were recently dewormed, simply because these factors could potentially affect the immune response as a whole. So looking at the objective 1, which was to determine the influence of helminth co-infection on COVID-19 clinical outcomes, we conducted a series of ELISAs, SARS-CoV-2 ELISAs, which was automated. That was done in Germany using the uranium analyzer 1. The helminth ELISAs were manually executed, and the results were analyzed using GraphPath Prism version 10. So within our study participants, gender was evenly distributed. The most participative age group were individuals aged between 11 and 22. However, individuals aged between 44 and 54 were less represented. Most of our participants were asymptomatic. We had very few severe cases. And the preliminary findings shows us that the helminth seroprevalence among our study cohort was 52.8%, and this prevalence is smaller than that obtained by Donko et al. In Ghana, and is higher than that obtained by Isomba et al. 2023 in Cameroon. However, the seroprevalence of helminthiasis within our study cohort was 41.3%, which is similar to that reported by Edward et al. 2024. Within the SARS-CoV-2 infected individuals, we found a helminth co-infection rate of about 43.6%, and within the helminth infected individual, we have a SARS-CoV-2 co-infection rate of 63.2%. And looking at the relationship between co-infection and the SARS-CoV-2 clinical outcomes, we found that individuals co-infected with helminth tend to exhibit asymptomatic to milder disease outcomes. However, individuals without Helminth co-infection tend to develop severe outcomes. More specifically, all the individuals with only SARS-CoV-2 infection tend to develop severe disease outcomes, suggesting that co-infection could actually be related to disease outcomes. Similarly, looking at the Helminth-specific antibodies, we found that the asymptomatic individual exhibited about 2 times more Helminth-specific IgG compared to the moderate to severe individuals, meaning that exposure to Helminth could actually be associated to asymptomatic disease outcome. Looking at our second objective, which was to determine the immunological basis between the 2 infections, we tried to conduct a series of ELISAs, SARS-CoV-2 neutralizing assays, and we also looked at 21 markers, 21 cytokines. And the findings were interesting. We found that individuals who were co-infected with Helminth and SARS-CoV-2 exhibited a significantly higher expression of type 2 anti-inflammatory cytokines. However, individuals who were infected only with SARS-CoV-2 tend to have more of the Th1 and the Th2, the Th1 and the Th17 pro-inflammatory cytokines, more specifically interferon gamma, IL-1 beta, IL-8, IL-2, IL-18, IP-10, IL-6, IL-17, 8. And similarly, comparing the cytokine expression within the various SARS-CoV-2 severity groups, we again found that asymptomatic individuals tend to have a higher expression of anti-inflammatory cytokines, more especially IL-5 and IL-10 and IL-5, IL-10 and IL-13, and they are known for their regulatory and their anti-inflammatory properties, which suggests that actually the higher expression of anti-inflammatory cytokines within this individual would have suppressed the action of pro-inflammatory cytokines, which would have led to the asymptomatic to mild disease outcomes. Similarly, the moderate to severe individuals showed a higher expression of pro-inflammatory cytokines. And with these results, we also went further to find out how co-infection could also affect the humoral immunity, the antibody immune response to SARS-CoV-2. And interestingly, we found that co-infected individuals tend to have a lower expression of SARS-CoV-2 specific Ig and IgG, which is actually in alignment with our previous findings due to the fact that if the co-infected individuals tend to have a suppressed Th1 immunity, it means that actually the Th1 immunity, which is responsible for antibody production, would also be impacted. Looking at the correlation, we also try to have a correlation matrix to see how the various parameters were interrelated. And we saw that Helminth specific IgG correlated inversely with SARS-CoV-2 specific Ig and IgG, and with the neutralizing potential against the Y type and the variants, meaning that as the Helminth specific antibody tend to increase, the neutralizing potential of against SARS-CoV-2 reduced, and the antibody response to SARS-CoV-2 also reduced. So looking at that objective, which was to determine the immunomodulatory effects of Helminth antigen on PBMCs in vitro, we isolated our PBMCs, we did our cell cultures, we stimulated them, we stained them with antibodies, and we did some FACS measurements. And with our gating strategy, we gated for all our cells. Within all our cells, we gated for the lymphocytes. Within the lymphocytes, we gated for the CD3 positive cells. When we had the CD3 positive cells, we gated for the CD4 positives and the CD8 positives. We're more interested into the CD4 positive, so we furthered our gating into the CD4 positive cells to get the interferon gamma producing cells and the CD154, which is the activation marker for SARS-CoV-2. However, these results are not yet available, and from the conclusion we could draw from the first 2 objectives could be that why the precise mechanism underlying this association requires further investigation. Our data provides valuable findings and statistical evidence supporting the hypothesis that Helminth co-infections confer some protective effects against severe SARS-CoV-2 outcomes by muting hyperinflammation associated with COVID-19 severity. And this hyperinflammation, which is mitigated via the type 2 pathway, may also simultaneously reduce the adaptive immunity against SARS-CoV-2 infections. Special thanks goes to the following institutions: for their funding and their supervisory support, GWAC, KNUST, KCCR, Bonn University Hospital, DAD, and the Bonn University Hospital. Thank you very much.
Let's do it better for NIMBOT. Very interesting study, and the immune modulatory role of helminth. So sometimes worms are good. I think that's what you are telling us, right? Yeah, worms are good. So we'll have opportunity to question NIMBOT later. NIMBOT, you should still sit in front. You've not had questions yet. All right. Our next presenter would be Mary Ifuakome. Mary is fourth year PhD public health, and she would be speaking to us on something very interesting. Let me be exact. Mary is speaking on the role of telemedicine for non-communicable disease care, an assessment of health worker capacity and patient willingness to use ICT for improved healthcare coverage in Ghana. Mary Comey.
Thank you very much. So good evening to everyone, and just a brief diversion from all the molecular analysis that we have been doing. We are seeking to look at chronic disease burden in Ghana and how best to ensure that we can increase access to care for our patients that are coming with non-communicable diseases. This will be the brief outline I would undertake. Just to emphasize that across the globe, it's been highlighted over and over again that there's a transitioning from the usual infectious diseases to the non-infectious diseases. Our non-communicable diseases are on the rise. And unfortunately for us in low and middle income countries, we are bearing the brunt of all these deaths. For Ghana, we are not spared as well. Our country profile for 2023 showed that 45% of all-cause mortality is currently attributable to non-communicable diseases, and these are largely the four main ones: hypertension, diabetes, cancers, and the chronic respiratory diseases. If you add injuries, it gets to 55% of all our deaths, and that's obviously a worry for us. But most importantly in here, for hypertension, which is our number one cause of CVD, cardiovascular diseases, we are diagnosing less than half of all cases. And the main worry has to be the proportion that are going undiagnosed. Now, these cases, when you look at it, it's no surprising that people are coming into the health facilities with strokes, heart attacks, amongst others. They are walking about undiagnosed, and accordingly, their conditions are not being addressed. Our health system has obvious challenges, key amongst them being the fact that even the health workers that are available are largely more distributed, highly concentrated, specialized services in specific centers, and the base population, which is in the communities, have very limited access to specialized care. Now, as we are seeking to improve on coverage of services, there's a need for us to do something different and look at more innovative strategies, not doing things routinely as we are sitting and waiting for patients to come to us, but then to reach out into the communities to address these conditions that are coming up largely without symptoms. Hence, the need to further look at how best to increase access using telemedicine and scaling up of the pilot projects, multiple pilot projects that have been undertaken across the country. Today's presentation will be largely on my objectives 3 and 4 for the main work, which are seeking to evaluate willingness of health workers to utilize telemedicine. Usually the assumption is that the health workers, they will use the ICT once it is made available. We actually wanted to find out from the health workers who were addressing the chronic disease patients as to whether they will be willing to use telemedicine for NCD care, and even for the patients as well, because there's a patient angle to telemedicine as well, whether they do have the capacity and also the intention to use telemedicine. So for the larger work, this was the conceptual framework, and it was considering the personal characteristics, system, technology issues, as well as operational factors, how it influences actual use, and ultimately how that also affects the decisions our policymakers take our health managers, the health workers in terms of their willingness to use, and then the persons living with NCDs. And it is the last two components on your right that I'll be presenting on today. So to undertake this work, we purposefully sampled health workers from hypertension and diabetes clinics. These were used as proxies for NCDs in 3 ecological zones of Ghana: Ghana East in Greater Accra, Amansia South, Ashanti, and then Bongo District in Upper East. And structured questionnaires were applied for this sample. We had a response rate of about 92%. Gender, there were more females, about 65% were females. And proportionate distribution across the regions using their regional level sample size as a baseline. For the health workers, willingness was measured using factors such as accessibility to smartphones, the ability to use computers, wearable devices, amongst others, their perception of usefulness of technology, as well as their personal experiences. And it was realized that 67.1% emphasized that they were willing to use telemedicine for NCD care. When you look at the usage of telemedicine itself, persons who had experience with use of computers were more positively associated with usage of telemedicine, as well as people who had personal innovativeness in that they were willing to try out new technologies. For the persons living with NCDs, 87.9% had the intention to use telemedicine, and this was, the various domains included perceived ease of use, perceived usefulness, as well as the behavioral intent to use. And then finally, in terms of capacity for the persons living with NCDs, it was determined by other factors such as ownership, availability, ability to search for information online, the understanding of information shared by phone, both audio and then video. So we considered telemedicine in the context of mHealth and video conferencing as well. And then there was a strong association between capacity and intentions to use when you considered them as binary variables. The chi-square was significant in that regard. So when the analysis was conducted for actual use of telemedicine, persons who said that they were not confident to use electronic devices were negatively associated with usage, whilst for operational processes, people who mentioned that they had support in terms of usage of telemedicine and could get enough information to guide them in their decision to use telemedicine were positively associated. But interestingly for me was the system challenges, which could potentially serve as obstacles in our implementation of telemedicine. All the system characteristics were significant. So it's critical for us that as we are seeking to increase access using telemedicine, we looked at internet coverage, access to electronic devices, and then our ability to utilize these devices, and then people's confidence in the system, which can either derail our efforts or promote what we are seeking to do. So finally, just to emphasize that for the objectives 2, 3, and 4, the health workers were willing to use telemedicine for NCD care in the selected districts. Persons living with NCDs who were patients from hypertension, diabetes clinics that were sampled had the capacity and intention to use telemedicine. And for policymakers, this emphasized the need to strongly advocate for alternatives to in-person visits. That as we are providing clinic visits, we should also strongly consider using telemedicine. And then experiences from COVID, as has been shared for us here, has shown that it works. So it's a good opportunity for us to consider telemedicine as we are considering addressing our increasing disease burden in terms of NCDs for Ghana. Thank you very much.
Thank you very much, Dr. Comey. And may I invite you and Kembot to the seats for the panelists. you are the policy maker, so I'll be interested in know how you utilize the results, because Dr. Kome is the lead for NCD services in Ghana. So I'll be asking you a few questions about that. Prof?
Thank you very much, Co-Chair. I'll take a first bite at this. Nimbot, interesting presentation, and thank you very much. Which other confounding factors did you take into account while I was doing this? Because generally, we know that COVID disease was very severe in older age groups. Most of the West, a lot of those who died were the aged, pension people. Okay. We don't have, the age structure here is different. Okay. So that for me could have been a confounder, why we didn't see so many people die here. So what were the confounders you used in saying that probably hermetic infections reduce our rates? And also, I get the impression that you were looking at people who use digital devices, who knew how to use digital devices, who were okay with digital devices. How is that different from telemedicine? Because my ability to use a digital device may not necessarily be my willingness to be involved in telemedicine.
We'll have you respond to the questions from the chair, and then afterwards we'll take from the audience.
Okay, Prof. Thank you very much for the question. Yeah, actually in our study, there's one, I just presented a summary of the results, because we also use a multivariate logistic regression to predict how co-infection or infectious status could influence the disease outcome. And in our model, we try also to screen out the effects of age, the effects of BMI. It's true we could not, from the data we have were limited, we could not screen so many confounders, but at least we screened for age and for BMI and for gender. And after screening out for these confounders, we found that Helminth's co-infection still had a significant effect on COVID-19 clinical outcomes. Thank you.
Yeah, Dr. Kome, and then we'll go to the audience.
Thank you very much, Prof. So for the health workers that were interviewed, almost 92% of them had smartphones. The remainder had the analog phones. For the persons living with NCDs, I was trying to avoid yam. For the persons living with NCDs, just about 2/3 of them had smartphones. There were a few of them who did not have phones personally, but relied on phones for their either caregivers or their children. The elderly women, they didn't have phones themselves. And then quite a big chunk also had the young phones as well. We were not looking at people who could use digital technologies, but in the midst of the clinic patients, how many of them were willing to use? And indeed, that was what we found out. So using digital technology already was not a prerequisite. However, the finding was that majority of them were already using mobile phones. And then our mobile phone usage as a country is about 133%. So some people have more than one phones as a country, and that is where we currently find ourselves. So most of our patients are carrying mobile phones already.
I think my question, how did you broadly define telemedicine? Because having a digital phone, being able to manipulate a digital device is not the same as telemedicine. So that's where my problem is. So now how do you broadly define telemedicine?
Thank you, Prof. So telemedicine was defined as providing healthcare using technology. And technology in this case could be a mobile device. It could be through teleconferencing, teleconsultations. It could be through blasting of health information to the mobile phones, just as the telecommunications networks undertake it. So once you were receiving some form of health service, but not directly sitting in front of, or not receiving it in person, but receiving it digitally, was considered as telemedicine.
We'll go to the audience, but to you, if I'm not sure if you assessed what your view is on the lack of content currently, because one of the drawbacks of this approach is fake news. You have people sharing all sorts of things to our patients. How might we have to address that? Audience, questions, show by hands. Yeah.
Yes, I would like to find out humbly from Doctor whether during our grandparents' era was telemedicine not in use, because it happened to appear that telemedicine has a link with ICT. So can you please expatiate that?
Yes.
Okay, I have two questions, and they all go to NAMBOT. So the first question is, you made mention of HERMINT, and we all know that HERMINT is very broad. So I would like to know the HERMINTs that you considered and what influenced your choice of those HERMINTs and how they were diagnosed. That's the first question. And the second question is, you all know that during the COVID-19 pandemic, the country was locked down. So how were you able to recruit the participants into your study?
Thank you. Is there any other question? All right, let's address these and then we'll come back to the audience. Ifwa and Nembot.
Okay, thank you very much. The question about whether telemedicine was used in the olden days, the history of telemedicine starts with NASA, who piloted sending messages into space and were hoping to get feedback. And as time went on, this sort of messaging was, should I say, modified, and sort of the healthcare system adapted it using ultrasound amongst others. The concept of telemedicine is looking at receiving healthcare at a distance. The tele component sort of introduces digital technology. In the olden days, people were carrying messages by foot. So when somebody is sick in the village, somebody can walk to the white man who has some kind of medicine at the shore, collect the medicine and come. I'm not sure if you can classify that as telemedicine. However, with how telemedicine is currently defined, the ICT component has to be present. I'm sure there are other modifications that can be looked at. Once the healthcare is not being delivered in person and the client is at a distance, the message is being conveyed. It's conveyed with the assistance of technology. It is considered as telemedicine. And then you mentioned content, fake news. It's a very real concern. For the patients, their main concerns had to do with service providers who were blasting messages, and the idea that these messages were not targeted. When it came to receiving targeted messages, I think almost, I would have to look at the data, over 90% were okay with receiving messages related to NCDs using telemedicine. But the concern had to do with they sitting there and arbitrarily receiving messages from telecom companies, advertising issues, that they had a huge issue with. But if the messaging had to do with reminder notices, reminding them about taking their medication, reminding them about their next appointments, clients were generally had a positive attitude and were more receptive. Indeed, they expressed the need for them to receive more messages in order to remind them. Because sometimes they did mention that aside the financial challenges. They would sometimes miss their appointment because they simply forgot when they had 3 monthly appointments or 6 monthly appointments. So if they could be reminded, they were very much receptive towards receiving these messages.
Just before you hand it over, in what languages were these messages being delivered? Because we didn't hear about that.
No, we did not implement telemedicine.
Okay. Right.
But the questionnaire was translated into Ga, Chi, and Dagomba. Yeah, so we did not implement the intervention. Yeah.
Okay, thank you very much. So for the first question, that of what are the health means, which I focus on in my research, I looked at Ascaris lumbricoides, I looked for Strongyloides rati, and Acanthochelonema vitae. And what informs my choice is that, you know, when it comes to helminths, there are blood and tissue, and we have intestinal. So first, I wanted to have at least a mixture of both tissue, blood and tissue helminths, and intestinal. We know Ascaris is more of intestinal, and with Strongyloides also, and Acanthochelonema is more of blood and tissue. The thing is, actually, Acanthochelonema vitae, it's not a human parasite, but the kit is commercially sold because when you want, if you want to cover a broad range of filaria nematodes, which are found in humans, they are known to cross-react with lymphatic, with Wuchereria bancrofti, Brugia malawi. They are known to cross-react with a lot of filaria nematodes, which are found in humans. So using the kits for Acanthochelonema vitae could actually enable us to cover a broad range of filaria nematodes, not just maybe Wuchereria or Brugia or whatsoever. So that is why we chose those helminths. And how were they diagnosed? We use enzyme-linked immunoassays. We use ELISAs because we do not have access to the stool samples to perform PCR. And with regards to the third question of how were the participants recruited, yeah, actually, as I said, this was a retrospective cohort approach. And these samples were collected at the Kumasi Center for Collaborative Research and Tropical Medicine. And that was one of the testing centers during the COVID-19 pandemic. So when individuals came for testing and they consented for their samples to be taken for a study, then that's how their samples got to be collected. So they were some of them were travelers, some of them were just people who just wanted to have themselves tested. So when they came to get themselves tested and consented to take part, their samples were taken. Thank you very much. I hope I answered your question.
Right. Thank you. There was a hand here. Show by hand up.
Thank you for the presentation. So I have a question for Nimbot. You made mention of interferon gamma and IL-6, 7, among others. I just want to know what is their role in the disease outcome you are looking at.
You have to ask 3 more questions, so otherwise we won't close. We need to get Prof. Ngala and Co. to score them on question answering. So, all right, Nimbot.
Okay, thank you very much for your question. So as of interleukin-6 and interferon gamma, they are type 1 pro-inflammatory cytokines. And as I said in my introduction, these type 1 pro-inflammatory cytokines, when they are being secreted in excess amount, they lead to the cytokine storm. The cytokine storm leads to the infiltration of these cytokines into the body organs. There's long into the lungs. There's lung inflammation, and that's what leads to severe COVID-19. So they have a very important, especially interferons, they have a very important role to play in mitigating the disease outcome, especially in the case of SARS-CoV-2. Thank you.
Thank you very much, Limbot. Any other question? All right.
Thank you so much, Limbot. I'm particular about the helmets you looked at. And you excuse me, I am not a medic. But if it is veterinary parasitology, if you are talking of helmets, hookworms is very, very important. Tapeworms are also very, very important. I was thinking that in looking at the kits, I don't know whether there are non-commercial available, especially for hookworm. If there is, then you should have looked at that also. Thank you. Good.
And as a clinician, I would say that even for humans, those are very important. So maybe it was due to kit availability. Any comment from you, Nimbot?
Yeah, thank you very much, Doc. Of course, you're very right. I think in our panel, we had to include some of these, like hookworm, as you rightly said, is one of the most common parasites. But in our settings, we had to limit ourselves with the kits that was available. Thank you very much.
Thank you very much, If you can resume your seats. Thank you. I think we've had very good presentations this afternoon. I certainly have enjoyed it, spanning the breadth of emerging infectious diseases, the viral ones importantly, some understanding about why Africa was not that much impacted with COVID, and eventually looking into the future with telemedicine. So, presenters, thank you very much. What I want to say, a bit of what the Provost Vice Chancellor mentioned that a bit in his presentation this morning, that the last year when OGR organized the Graduate Students Conference, I don't know if you remember, those who were about getting their theses ready, etc., were able to work with the supervisors, and they submitted some manuscripts to just the University Journal of Science and Technology. And about 10 of them have gone through the review process, and they will shortly be published. Just as our own journal, it is there for you. It goes through a peer review process, a very good one. And so I acknowledge some of you and your supervisors, those of you who have some data and who are ready to want to publish, please have a look at just more so as the graduate school is coming to say that you need a certain number of publications as part of your PhD. So as you consider publishing, consider about publishing the Journal of Science and Technology. So on behalf of the organizers of the conference, I want to thank you, the audience, you're a very good audience, and the presenters as well for your attendance at the session. I'll give the final word to make some announcements.
Not the final word, because we are in Ashanti, so when the Ohene speaks, nobody speaks after. I cannot be speaking after my immediate past provost, but Prof, thank you. It's been wonderful doing this with you. And thank you, the audience, too, for being such wonderful audience. I think you deserve a clap, so you should clap for yourself. Yeah. We have a second session tomorrow, and I've been alerted by the conference chair, Prof. Deborah, that our registration starts at 8:30, and then the actual panel sessions begins at 9. Please, let's do well to be present on time, and we look forward to seeing you here, beautiful people, tomorrow.
So thank you all for coming. And
you have a chat.
Yeah.
This question is above me.
Yeah, so it's above your pay grade.
It's above my pay grade. So you can have a conversation with the conference chair, Prof. Deborah. But as far as I'm aware, this is a college-approved program. So have a session.
So we hope to see all of us here tomorrow morning.
Yeah.
So have a good evening.
Have a good evening. Thank you for coming.